Hello, my name is Cole, and I'll be talking about glutamine transporters and cancer cell reprogramming from the paper that I read. So the paper that I read is a variant of SLC1A5, is a mitochondrial glutamine transporter for metabolic reprogramming in cancer cells, made by these people. So we've actually mentioned this in passing back when we talked about amino acid catabolism. That's where this picture comes from, is from this article, and it was actually in our slides. So as you can see, it's just kind of the overall abstract of what they're doing. They're talking about this variant and how it transports glutamine into the mitochondria to do other things. So what is this variant? Of course, because it's a variant, it's on the same gene as the regular SLC1A5, but it's just transcribed at a different part of the gene. And so this will be important for later. These are also mostly found in cancers, especially common in very common cancers such as prostate, liver, colon, lungs, and even the pancreas, and a few other ones as well. But in this experiment, I think they specifically test the pancreas cancer cells. So a big part of their experiment was trying to find out what exactly SLC1A5 variant, like where it's supposed to go. And so they did a few tests. They did some subcellular fractions, so they basically just broke up the cell to see where this protein may end up going. So they tagged it with a bunch of different metabolites and to see how it would where it would bind to. And so in these images up here, G and H, it kind of shows some electrophoresis. So the darker bands means that they are more present there. So you can see that they're a little present in the homogenate. Of course, that's just kind of the whole cell broken up. Not as much in the cytoplasm, a little bit in the plasma membrane, and then there's a good chunk in the mitochondria. So they went into this further, and they found that they're more specifically found in the inner membrane of the mitochondria, which is that's what IM stands for. And they also showed that this is indeed involved in glutamine processing because as you can see in the black, that's the variant. You can see there's a much higher intake of glutamine with this variant. And then if you inhibit it, which is what this SI means, there's a much lower amount of glutamine that shows up, and the other ones have to make up for it. But even then, they don't do as much because, as you can see on this part of the graph, it only goes up to 100, whereas usually it can go up to 200 with that variant. And so this kind of just shows that this is overall a mitochondrial transporter for glutamine, and that was kind of what they found out at first. And so after that, they came up with a structure for it. This is kind of what it looks like. And it's important to kind of pay attention to where these N-terminus and C-terminus are. I'll talk about them more in a bit, but this is just kind of the overall structure of what it kind of looks like. Kind of goes in through the membranes and everything of the mitochondria. And of course, this intermembrane space is kind of important. We've mentioned it before. It's very different in pH and charge compared to the matrix. It's, of course, more positive, where the matrix is more negative. So these could be important. And so coming back to those C and N termini, they need to find out how it gets to the mitochondria in the first place. So it's trying to find the target sequence. They believed it to be these C and N termini because they do end up in the inner mitochondrial membrane space. So what they did was they just tagged them to see if they actually showed up there, and they tagged them along with cytochrome oxidase 4, which, as we know from class, is really important in aerobic metabolism and respiration, because it's like the last step in the respiration before it gets to ATP synthase. And so these are actually co-localized together because they're both in the membrane and they're stuck in it. So of course they're going to end up in the same kind of places. So it kind of shows you where they are. Here's the mitochondria. The green shows you the variant. The red shows you the cytochrome oxidase. And when they're unimpeded, they end up in kind of this membrane space, a little bit outwards at some points, but for the most part, they're very together in where they're supposed to be. However, when you mutate these C and N parts, they end up kind of falling away and going towards the cytoplasm. So as you can see, this one's a lot more orderly. You can see the red and green kind of mixed together into yellow. Here, it kind of looks like you took a gusher and stepped on it and it kind of splurried everywhere, and there's a lot less order. So it's definitely involved in targeting the mitochondrial membrane, and so they believe it to have that kind of, they believe it to have a targeting sequence to get there in the first place. And so enough about the structure of it. Let's actually talk about what does this do. And so this comes into the reprogramming aspect. They found it can be induced by hypoxia, which is important because cancer cells put a lot of stress on oxygen transport because they grow so fast. We've mentioned it in class before. They often work in hypoxic conditions because of all the stress that they put on the body. And so they tested for this. So as you can see in their charts, N stands for pneumoxia, so this is just normal oxygen levels, and H stands for, of course, hypoxia. With the variant around, and also its regular one as well, they are around much more in hypoxic conditions, at least their mRNA levels are, showing that they are important to hypoxia and that these can help possibly to upregulate protein for more glutamine uptake for a bunch of different things that we'll get into later, such as metabolite synthesis and ATP. And before we get into that, I want to mention just something else we mentioned kind of way back in class, which was hypoxia-induced factors. When we talked about hypoxia, of course, these are activated by these conditions. How is the cell going to deal with them? Because it's not great when you don't have any oxygen to make the most amount of energy. We mentioned HIV-1, and so that usually uptakes the non-aerobic mechanisms. So there's more glucose, there's more fermentation with the lactate dehydrogenase, and the other aerobic cycles like TCA are kind of turned off and not really focused on as much. Or, of course, they can just replace parts of the aerobic respiration to work a little bit better. And so these usually are a lot better in these hypoxic conditions. But what's weird is this variant and also its regular version don't really seem to be affected by HIV-1 like we've mentioned before, because when they tried to downregulate it, it didn't really change their expression. However, HIV-2 alpha, when it was overexpressed, a lot more of these proteins showed up, and of course when it was underexpressed, the opposite was true. As you can see in their data here, they had a control group just kind of showing that when they put in HIV-2 alpha, it didn't really show up as much, but when they didn't put it in, it was just the regular conditions, but when they overexpressed it, these showed up a lot more. As you can see, they're very faint, but then they get a lot darker and thicker, showing there's a lot more there, a lot more protein that shows up in the gel. And they also did a bit of a amplicon test. So this is basically just to see, okay, where, if at all, does the HIV bind to and to turn this on. And so they found it's mostly in amplicon 1. As you can see this big bright thing right here, and that is going to be around here, which is somewhere in the promoter for these genes. So of course, this is definitely going to express it during hypoxic conditions, and that shows how it can help reprogram the cell and get upregulated for transcription. So enough about HIV, let's talk about glutamine, because that's what it's bringing into the cell, and we got to see what it does. So first and foremost, when we think about hypoxic conditions, we're automatically thinking, okay, there's less energy, how are they going to get it? We usually think of glucose and it's going into fermentation, and so that is what we usually get. But what we find is that in hypoxia, it is really important for glutamine to be used because it can actually be useful for ATP generation. As you can see in these tables, when glutamine is present, if it's under normal conditions and there's glucose around, it's kind of preferred. When glucose isn't present, glutamine is still important to get this ATP. There's still a good amount of ATP being made. And it's really important when we start talking about the actual protein. So when it's turned off, you have these kind of lower amounts of ATP. But when it's turned back on, you get much higher ATP, showing that it's important to get this glutamine in. Of course, you can see it right here. It's a lot higher than it was over here when it was turned off. And so glutamine can produce some ATP from different processes. Of course, when it can turn into glutamate, and then that glutamate can turn into alpha-ketoglutarate through some transaminase reactions that can make some ATP. But what else can it lead to? So like I said before, it is, like we've kind of mentioned and kind of may think about, we usually think about the urea cycle when we talk about the glutaminolysis. That's a hard word to say. It's going to, take glutamate from other places, turn it into glutamine to be transported. Once it gets into mitochondria, turn to L-glutamate, and then it's going to go off, as you can see, probably to make urea. But it can also be turned into different things. Like I mentioned before, it enters the TCA cycle as alpha-ketoglutarate, and they know that it actually affects these cycles because when it's turned off or severely decreased, these metabolites also decrease with it. So as you can see, This Si, of course, again, means that it's turned off. When it's turned off, there's a very low amount of alpha-ketoglutarate. And in subsequent TCA metabolites, such as succinate, that's low as well. Fumarate, malate, all of them are low. Not great. And this is really important because these metabolites are really important for other redox reactions, not only in the TCA, but also for lipid synthesis and a few other processes, which we'll get into soon. Another thing is, we mentioned also in class for a little bit, glutathione. Now, we haven't really gotten into this yet because we haven't gotten into nucleic acid synthesis, but we will soon. But when you take glutamine and turn it into glutamate, it can also be turned into something called glutathione, which we have mentioned before as glutathione peroxidase, which helps deal with reactive oxygen species. Now, we've been talking about hypoxic conditions, but soon I'll actually show that in this paper they talk about aerobic conditions as well. So this peroxidase is actually really important to upregulate, and I'll show you why in a second. But this is another thing that glutamine can lead to, and it's very helpful for a variety of reasons, especially survivability, because when you're a cell, you don't really want to deal with these things because reactive oxygen species, like we mentioned before, can mess up a lot of things like DNA, and while that can lead to more mutations, it can also just straight up kill the cell. You don't want that. So let's talk about some other metabolism. So as I mentioned before, glutathione can be made. This can actually help out with the pentose phosphate pathway. That was mentioned a while back. And so this can kind of help recycle the NADPH, and you can actually get more of these intermediates. So ribulose 5-phosphate will eventually be made after you kind of recycle it with this. And then that will give you recycled back into glucose 6-phosphate. And then that can go into glycolysis, which is really important. And then, so you actually have two things going on here. You have the PB pathway and then the glycolysis. There's also, of course, biosynthesis. So these can be made into fatty acids and sterols and everything. And that's not all, because you also have the TCA cycle that I mentioned before. Of course, you enter alpha-ketoglutarate. You can go through the whole TCA cycle for energy, or you can also have citrate, which is good for fatty acid synthesis, as it's a good precursor. And still going back to the pentose phosphate pathway, this of course leads to nucleotides, which is also good for nucleic acid synthesis. So glutamine is actually super duper important to all of this. The paper also mentions fructose 1,6-bisphosphate and lactate, which can also be used in the glycolysis pathway. So again, it's just a good just do all. But that's not all the metabolism that it can do. Of course, using the TCA cycle, you're going to get a lot of good fuel for aerobic cell respiration. So they actually found that the SLC A5 or SLC 1A5 is actually really good. Sorry, there's a typo there. That's supposed to be 1. But it's actually really good for aerobic metabolism as well, not just hypoxia. As you can see, they measured oxygen uptake and use. So they found that it is used a lot more when SLC 1A5 is around, and they labeled these as, glutamine and glucose being, or this actually not glucose, this is glutamate. You can see on the red line that it goes up a lot as time goes on. It is taking up a lot of this oxygen, meaning that it is very involved in the in using oxygen for this cellular respiration. And glutamate actually isn't as important. It seems that glutamine is a lot more important to this than glutamate is, for a variety of reasons, mostly because glutamine is a lot higher in abundance. It's one of the higher amino acids in the blood, so it's very easy to use for things, while glutamate can kind of get used up pretty quickly. But that's not all, because it provides that glutamate, of course, can lead to alpha-ketoglutarate and all that for the TCA cycle, provides all of those electron carriers that we need for the respiration. You can also get succinate out of a TCA cycle, and that can farm through the complex 2 to help out with also generating all of these protons and that proton gradient, which can of course get us our energy. And this is all upregulated by this protein, which makes it super duper good for getting energy for these cancer cells to continue growing. And so that's kind of the big thing about metabolism that can connect to class, like what we talked about with all this stuff. But I kind of want to mention one more thing they talk about, which is the gemcitabine resistance. I don't know if I'm saying that right. This is just a chemotherapy drug mentioned. It's kind of used fairly often, and it's effective because it's an anti-metabolite, gets in the way of making DNA and RNA. Of course, you need that when you're a growing cell. So what they found is, in previous studies as well, cancer cells can kind of confer resistance to this in hypoxic conditions, making it less effective. And like we mentioned before, SLC1A5 is activated by hypoxic conditions. As you can see in these graphs, they actually have a higher rate of survival, while it might not look like it at first. They start higher because the hypoxic conditions are going to kill a few of the cancer cells. They're going to start lower, but they have a very gradual decline when gemcitabine is added. In normoxia, they almost go down immediately, showing that their average effect of growing rate is way lower, showing that this is very effective. And then, as well as over here, you can see it as well. The hypoxia is a lot better at it because it has a much larger infection, growth infection ratio. When it takes a lot more cells to actually be affected by this growth, that means it is incurring resistance because it's not working as well. There's still a lot of growth still going on despite this drug being used. And so why do we think it inhibits the gemcitabine? I'm not really, my knowledge is not that far in, but the paper does mention that this could be due to glutathione synthesis and pyrimidine synthesis. So the glutathione synthesis can help with dealing with reactive oxygen species, and as we mentioned before, the pentose phosphate pathway is upregulated. That leads to a lot more nucleotides. There can be a competition and between these two things where it's making so many nucleotides that the drug doesn't really do its job in inhibiting it. It can just keep making enough to actually just go past it and doesn't even have to worry about it. And so this is actually really important to talk about because that leads to another way that cancer can be dealt with if we actually get rid of this. As you can see in these, when it's turned off, a lot more cells die, but when it's left on, then not as many cells die. You have around 200 over here when gemcitabine is in there, as well as this other thing, this BSO thing, this BSO molecule, my bad. It restricts the glutathione synthesis, and so it actually kind of incurs more death. It actually makes this more effective again. So when you remove it, it can actually help with making this drug more effective. So that leads me kind of to the end of the study. So why is all this important? Well, of course, it provides a new avenue of cancer research past just the regular gene that the many other studies have mentioned, because it shows variation to be much more important to cell growth. When this variation is made, of course, it's a lot better in hypoxic conditions, better for glutamine transport, which, as you can clearly see, relates to so many of the metabolic processes that we've talked about before. And it is, of course, like I mentioned, a very good target for anti-cancer drugs, because if you can turn it off, it shows that previous drugs have a higher higher effectiveness than they would have with these on. And so that leads me to the very end with my quiz questions. A lot of them go over mostly just kind of how it connects to different cycles. Thank you for watching, and hope these questions aren't too hard.